My name is Jana Bromberg, and I'm going to talk to you a little about what my lab does and then how I see this fitting into the preventing pandemics theme. So the main research that we do, the main focus of the lab is on molecular functionality. So we analyze genes and genomes and protein structure, protein function, and how they add into systems. And we do this all in a dry lab space. So that means that we use a lot of machine learning, hence the wonderful picture of the brain that doesn't quite look like a brain over here. And we also do some network analysis and all other neat things. So I'm going to talk a lot about this piece over here. I'm going to start with talking about microbes and microbiome. So what is a microbiome? Microbiome is a collection of microbes in a particular niche or environment. So you could sample microbes from the ocean, or you could sample microbes from the soil, or even from the gut of animals and these funky little baby over here. So the problem with studying microbes the way that I would like to study them, so via the genetic material, is that collecting the individual genomes of the bacteria that are present in these microbiomes is extremely difficult. So if you think about it, this is what it would entail. You would sample some DNA from a particular niche, environmental niche, and you would presume that it has a bunch of bacteria in the genome, but their genomes can be extracted. But you don't know what these bacteria are, nor do you have a way of separating one type of bacteria from another. So what happens is you cut these into really small fragments, and then you try to sequence that, because that's how our sequencing works right now. But once you've sequenced it, you would then need to try to assemble it in order to figure out who is there. So if I was going to give you an analogy, think about this wonderful and beautiful college library. This is Trinity College in Dublin. It's really an amazing place. And if I ask you what are the books that are present in this library, what you would go out and do is shred every single book, get a little scanner that tells you what is the sentence that's written on that particular shred, and then try to assemble this using large-scale computing to figure out what books would have actually been there. Now, of course, you're not going to get very many of them correctly. They're also going to come out in a weird shape and may have information from other books accidentally pasted in. But in the end, what you will get is the bacteria that are most likely in this environment, or the books that are most common in this environment, and not anybody else. So we thought that this was overkill in terms of having to assemble the genomes, and we could potentially get the information that we were interested in, that is the molecular functionality, using direct annotation of reads. Now, I have to say that we are not the only ones to do this. There are many people who do this as well. But we thought that our method, we had a great idea for making it super precise, and this is how we did it. We call the method MyPhaser, that stands for Functional Annotation of Sequencing Reads, or Microbial Functional Annotation of Sequencing Reads. We collected the bacterial genomes for which we had annotation already. And what you see here is an EC number, an enzyme commission number, which is a hierarchical way of representing, of annotating the functionality of a particular protein, so a particular enzyme. So let's say 1 over here stands for oxidoreductase, and as you go down in the digits, you will get more and more precise in annotating functionality. So we have these reference proteins from the bacteria that have been studied experimentally. And we also had the genomes of those same bacteria. So what we did is we took the genome information from these bacteria and had created cuts to produce reads or read length sequences for every single one of these genomes. And then for every read, we applied 6-frame translation to get this to protein state, because we're mapping them back to protein. So the way that our method then worked is by taking the alignment, sequence alignment of the peptide from this read to the reference protein of a different bacterium. And what we looked for is that the annotation of this reference read was the same as the one that came in the aligned protein. So you see this blue dot over here, that indicates that this is correct. So it came from one bacterium to another bacterium, alignment of the read from one bacterium to the protein of another bacterium, and they share annotations of the functionality. And what you see here is that this, for instance, would be incorrect because the function here is 2.3.1.5, and here is 1.1.2.1. But you could have the alignment anyway. So we did this for a large set of proteins and the randomly generated reads that we had. And we plotted them into a space of alignment links versus sequence identity. And what we very clearly saw here is that the majority of the correct alignment, that is indicating that a read has a particular functionality that belongs to that specific protein, is actually up in this upper hand corner over here. And we were able to optimize a gradient decay curve fitted through this space, which would separate these blue dots of the correct alignment from the red dots of the potentially or putatively incorrect alignment. And so it was a fairly simple optimization problem for this gradient decay curve, where we just optimized the two parameters, this exponent over here and this multiplier of the length. So this method then could be used for predicting the functionality of the new read, as long as this functionality was actually represented by our reference protein. What we found is that MyPhaser outperformed BLAST, which is a basic way that you could do the same thing, by a lot. So MyPhaser was a lot more precise. And in fact, the thing that you see here is a very commonly used cutoff of BLAST of 10 to the negative 3 is actually doing better than MyPhaser in this level of resolution, where precision is about 50%. So about half of the annotated functions are incorrect. So we have this wonderful tool. What can we do with it? Well, think about it this way. Let's say you come in with a metagenome data, that's a lot of reads, and you can annotate them according to the functionality that we have in our reference database of the proteins. So then your metagenome is a vector of functions annotated with the abundance of the reads that map to each functionality. And then you can compare these vectors to each other in any way you would like. So one approach is to use a Jacquard distance between the individual reads. So, I'm sorry, between the individual vectors describing metagenomic data, which is what we did here. Now here, the metagenomic data comes from the BP oil spill. That indicates the, so this particular experiment was done on the beach sand. So we collected, or people have collected microbiota from the Gulf beach and then measured the difference between the pre-oil phase, the time during the oil phase, and the post-oil phase. And what you could see very clearly here is that the vectors of functions of the bacteria that live in the oil time separate really well from the post-oil and the pre-oil phase. So this is a tree that you would build for hierarchical clustering. So this is one cluster, and the red and the green are another cluster. So first things first, the oil space separates really far away from the post-oil and the pre-oil space. Another thing that we could do is potentially look at these vectors mapped into a reduced dimensionality space, like here. This is the non-metric multidimensional scaling, and what you see here is two-dimensional representation of all the samples that we had for the different phases of the BP oil spill. What you see here is the samples that came from the pre-oil spill. The dark black circles over here and rhombi indicate the oil times, and this is the squares that are post-oil. Now, it could be my imagination, but one can definitely see how there is recovery happening over time, in the sense that this post-oil spill microbiome actually looks like it's migrating closer to where it had been pre-oil spill. Now, we could apply the same type of techniques to figuring out whether individuals have a disease or they don't have a disease. And we also see that this matters in terms of the host sex and the generation of the host. We had a fairly small sample here. We had this family. This is a family of people who are related to Crohn's disease patients. So the first thing that you see here is that the Crohn's disease patients are indeed very far separated from the healthy population of their family, even those that live in the same household and eat presumably the same food. But one more thing to point out here is that Crohn's disease patients tend to have different functionality of their microbiome. The length of the branch here indicates that, and you could see that they're fairly far apart from each other as well. There are some other things that you can trace here. So for instance, if you look at the closest couple over here, those are two first generation people. It's a couple that lives in the same household. And interestingly, they share the same microbiome, that's as expected, but their closest microbiome relative is this child over here. Now, this child is not even a direct descendant, right? So it's a step away from these people over here, but she is very close to them. And part of the reason I think that they could be close is because her mom seems to be microbially very close to the child living in this household of the original couple. One thing to notice here is that in this generation, there are two healthy women, one sick woman and one sick man. And the sick people we already talked about, but these two women that are in this generation, so SO3 and S12, are actually not related to each other. So they're related by marriage, but they share the same microbiome or very similar microbiome. And that speaks for generational and potentially sex difference in microbiomes, because the healthy male in this population, SO6, is actually far removed from these individuals that are close to each other. Now, another thing to confirm that generation and sex matters, there are these two female children at SO5 and S10, and they are very close to each other as well. And again, the outlier boy, SO4, is far away from everybody else. So this type of tracing is something that you can do with the microbiome data, and it's very reassuring to see things like this, because it means that the signal that we are picking up is actually meaningful to the specific environmental niche that the microbiomes reside in. So furthermore, by looking at the healthy sick people, so this Crohn's disease versus healthy people over here, we can evaluate what are the exact functions that actually contributed to that differentiation. There's a lot of them, but the two that stand out the most are lipopolysaccharide biosynthesis and glutathione metabolism. And these are microbial functions that actually help bacteria promote inflammation and help it survive inflammation. So this is a very interesting way of looking at how the individual functions actually affect the particular phenotype of the individual that carries the microbiome. We also saw that the diversity of the microbiome corresponded to the overall well-being of the individual. So here we're looking at the microbiota that comes from children that have Prader-Willi syndrome. They happen to be obese. They're being put on a diet. And at day 0, their diversity is minimal, so they all look fairly similar. On the other hand, as intervention proceeds, the diversity of their functional diversity, of the functions that are available to their microbiota, increase. And that's an interesting observation, which actually corresponds to what a lot of taxonomic study says, that microbiota diversify with improvements in health. So another thing that we could look at is a group of people that have type 2 diabetes versus healthy controls. And we can train a machine learning model to separate the vectors of microbiome functions of the healthy individuals from these sick individuals with a fairly good accuracy of the ROC area under the curve 0.74. And I think, again, a really important point here is that we can actually put a name to the pathway that is important to do this separation. So we're talking here about these two pathways that are involved in sugar metabolism, which is actually something that you would expect from the type 2 diabetics. So these are all these really cool things that you can do. And here's an important note. We have a website where you can go and upload your metagenomic data from whatever it is that you're looking at. And we're going to give you that vector of functions that you can then explore in any way you want. This takes almost no time. It's really fast, so it compares well with existing methods that are out there. So this is an important point. The fact that we have this web service is actually important. I'm going to come back to it in a little bit. Okay, so we have this method of identifying functional functions encoded in the metagenomic data. Recently, a postdoc of mine had decided to try something else. And what she did is she trained a language model. It's called Looking Glass. She called it Looking Glass. It's an LSTM. And the idea here was, can I predict the next nucleotide in the sequence? Now, this is a fairly silly task. And the reason that we had decided to do this is because that she wanted to create an embedding of the reads, which was not simply the letter representation that we're so used to seeing. So the idea was, let me train the machine learning device, or recurrent neural network in this case, to recognize the next letter. It will learn the language of prokaryotic genomes, and then we can use it in order to ask more complicated questions. So you could see that the performance of this model never gets off the ground, so it's about 40%. It's not random, because predicting the next nucleotide is definitely not as easy as 40% accuracy would suggest. But she trains this thing, and then that means by the end of the training, we have a way of embedding the nucleotide data from the reads into this 104-dimensional space that she comes up with. And once we've trained that, we can ask the model different questions. So we can train the model, for instance, to recognize the function of the particular read. So we asked a very generic question, is this read an oxidoreductase or coming from an oxidoreductase or not? And the idea here is that oxidoreductase is a fairly high-level prediction. So remember those EC numbers? That's the first number. And so it should be a fairly simple task, except our reference database is fairly limited. So when you ask MyPhaser to do this, it's only able to identify a fairly small amount, or actually a very small amount, of the reads as belonging to this oxidoreductase class or not belonging to the oxidoreductase class. If you ask MGRAST, which is another very popular method for annotating sequence reads, it's actually only able to annotate about 30% of the data. So why is that happening? Well, it's happening because the underpinning there is that you have to have a reference protein or reference gene to which to compare. And that sort of kills discovery, right? So for anything that doesn't look like something that you have, you cannot make a prediction, except if you use this embedding method, embedding-based method that we have created. And what you see here, of course, is that you can annotate all 100% of the reads. Actually, 20% of the reads happen to be oxidoreductases or belong to genes that are oxidoreductases. And then, of course, you can ask the question, well, how accurate is this? Because you can annotate everything just randomly. And we can't really check it, but we compared our expectations to the results that we have here for a data set of microbiomes where we knew how many oxidoreductases to expect, or rather, what is the relationship. So at the surface of the ocean, we expected fewer oxidoreductases than in the oxygen minimum zone, which is what we see here, and the mesopelagic halfway zones are actually also identified where they're supposed to be. So this type of ladder that you're seeing here is actually not visible for either MyPhaser or MGRS, probably because of the limited amount of data that is actually annotated. So the next question is, and that's the most important question to this particular audience, is can we learn the viral language that describes pathogenicity? So if I were to take these viruses and then I was to get the DNA or RNA data of these viruses and then train a language model, just like I did for the prokaryotic things, then I could potentially have embeddings of the reads that come from these viruses. Another thing that I could have from these viruses is annotation of whether the virus is humanly pathogenic or not. And then I would have embeddings of the reads and the annotation of the virus as pathogenic or not on the basis of the virus that they came from. So here, the next question would be, can I train a model, just like we did for oxidoreductase, of whether this particular read is pathogenic or not? And I could, and in that case, any new sequence that I see could then be annotated as being pathogenic or not. And this is an important point because where would I get these sequences? Well, there are two ways to get them. One is you could potentially just hunt for them in whatever microbiomes that you would like to sample, some forest microbiome or city microbiome, go swap down the subways like we've been doing and look for that. Another way is to use that interface that I had talked about where people submit microbiomes for annotation and see if we can identify any pathogenic reads in those data that people submit for other reasons. And this is actually an interesting point because you would have access to a lot more microbiome data than you otherwise could. The only catch there is that you would then need these people to actually alert the authorities in case they discover some pathogenic viral load. So that's the main idea that I wanted to talk to you about. But there are a couple of more things that I wanted to mention. One is this evaluation of the effect of the variation that inevitably happens in viral evolution. So there are very many methods out there that predict the effect of variation. So we're looking at non-synonymous variant effects. So this is basically the variants that change the function, they change the sequence of the amino acid. So the protein changes as opposed to just nucleotide changes. We have a method among these, it's called SNAP. SNAP is a neural network-based method that's been around for quite a few years. And it's interesting in the sense that it makes predictions of the functional effects of the variant as opposed to any human relevant pathogenicity or anything like that. So it's literally making a prediction of the effect of the mutation. And this is not our work, but someone has taken the SARS-CoV-2 spike protein and evaluated it using SNAP. We know that for a full set of all possible variants of the spike protein, 52% have an effect. And curiously, of the variants that are observed, so of the strains of SARS that we see, only 25% have an effect. So an effect could be deleterious, in which case it would probably not persist in the viral population, or it could be beneficial, and that's how the virus would evolve. So you could look for those over here, the beneficial ones that make this particular strain better. So the questions that we can ask here is, can we predict novel viral strain threats on the basis of the entire set of variants that are possible, that are going to have an effect in this proteins of this particular virus? And also maybe we can evaluate if the old treatments will be effective, because if the protein is completely changed, maybe the old treatments will not be effective. But if it is kind of the same and it's active about the same way and has kind of the same structure, then maybe we can see that the old treatments will work. So now another thing that I wanted to mention is we recently built this model, SynVAP, that identifies functional effects of human synonymous variants. So there are also other methods to do this. We claim that our method does best because it is able to separate the effects versus the benign variation. But I think the main point here is that we trained SynVAP with no human data available. So there is no data, experimentally available data, or not enough experimentally determined data of the effect of synonymous variants. So what we did is we just used observed versus non-observed variation. And we can do that for new viruses as well. So the question here is, can we predict effects of viral synonymous variants on the basis of the things that we observed, or the observed strains? And I think that's where I'm gonna stop. So I have only questions, no answers for you. Can we build a sequence-based pathogenicity predictor? Can we annotate the functional effects of viral variants to predict evolutionary trajectory? And also, can we explore the human genome? So I didn't talk about this at all, but we do this a lot. Can we explore the human genome variation for signatures of viral susceptibility and or for potential druggable pathways? So that's about it. These are all the people that do the work. This is Adrian. She built the LSTM that I talked about. This is Ishwa. He had built this anonymous variant effect predictor. Chen Sheng and Max built the MyPhaser method, and all the others have participated very closely. This is our funding, and thank you.